Hello everyone, I'm Shuze Wang. I'm a third-year PhD student at University of Michigan. Today I'm going to talk about mapping the regulatory landscape of auditory hair cells from single-cell multi-omics data. This project has been accepted by Genome Research. If you are interested in our project, please feel free to take a look at our manuscript. Hearing is mediated by the organ of Corti, which is within the cochlea of the inner ear. The organ of Corti consists of two major cell types, sensory hair cells and supporting cells, that originate from a shared progenitor. The two major cell types can be subdivided into inner hair cells and outer hair cells, and a number of different supporting cell types including pillar cells and data cells. Interestingly, tight spatial temporal control is required for development of the organ of Corti, which occurs in waves both molecularly and functionally. However, gene regulation mechanisms of the cochlear development are not fully understood. Weddington's epigenetic landscape is a metaphor for how gene regulation modulates development. During differentiation, a cell corresponds to a ball rolling down a hill, while epigenetics correspond to the landscape and affect the cell differentiation, and the strings shaping the landscape correspond to transcription factors. Our goal is to identify the regulatory landscape controlling the cell differentiation and maturation of the organ of Corti. To resolve our goal, we applied single-cell ataxic technology. In order to enrich for supporting cells and hair cells, we use the ATO1 GFP allele in combination with FGFR3 TD tomato lineage tracing. At postnatal day 2, cochlear ducts were divided into apical and basal compartments. Then we used FACS to isolate individual cells and specialized hair cells and supporting cells. We sequenced around 2,000 cells with median unique fragment count of 20K per cell. After initial quality control, we identified 6 clusters using snap attack method. We generated an age-matched single-cell RNA-seq dataset using the same mouse model. Flow gates were set a little bit less conservative, therefore we include more cell types than we expected. We sequenced 1,000 cells with a median unique count of 12K reads per cell, and we identified 11 clusters in total using Seurat. We used the canonical gene markers to determine the cell identities for single-cell RNA-seq data. To identify the cell types for single-cell ataxic data, we calculated a G-card index similarity matrix, which compares the similarities between differentially expressed genes from single-cell RNA-seq and annotated differentially accessible regions from single-cell ataxic. The rather the more similarity between two datasets. For example, cluster 1 in single-cell ataxic has the most similarity with hair cell cluster from single-cell RNA-seq dataset, and cluster 2 is more similar with supporting cell cluster from single-cell RNA-seq data. The first question we were trying to ask was that can we estimate transcription factor activities? To be more specific, can we identify critical transcription factors of hair cells and supporting cells, and then classify them into activators and repressors based on their functions? To infer transcription factor activities, we use the ChromVAR, which is an existing R package estimating transcription factor associated accessibility from single-cell epigenomics data. ChromVAR calculates a normalized TF motif activity Z-score for each TF motif. To determine critical transcription factors of hair cells and supporting cells, we identified cell type-specific transcription factor genes and TF motifs between hair cells and supporting cells. Next, we classify the TF mode into activators and repressors based on two assumptions. First, upon binding of an activator TF, chromatin accessibility at the regulatory element is increased, resulting in an upregulation of the respective target gene transcripts. And conversely, Binding of a repressor TF decreases average chromatin accessibility as a regulatory element, and downregulation of the target gene will occur. These two assumptions were adopted from diffTF, an existing algorithm. Based on the two assumptions, we established a classification model by considering the relationship between the TF gene expression level and TF motif accessibility for each individual TF. Activating TF function, resulting in chromatin opening, was inferred if a given TF was differentially expressed in one of the two populations, and the corresponding motif was significantly accessible in the same population. Like ATA1 and LHX3, it's shown in the first and third quadrants. A repressor was classified based on differential expression in combination with a negative correlated z-scores. The repressor was shown in second and fourth quadrants. The classification results were shown in this dot plot. Activators are classified in green, repressors in red, and undetermined TF is in gray. Overall, we identified 56 activators and 23 repressors, including lots of known TFs in the organ of Corti. To quantitatively validate our classification method, we compared the overlaps between diffTF and our method. The Venn diagram shows the number of overlaps between two methods. For activators, there are 26 overlapped TFs. For repressors, 7 TFs are overlapped, and in total, 33 TFs are overlapped between two methods. And the overlap are significant by conducting hypergeometric tests. Our next question is how these TFs regulate downstream target genes driving the maturation of hair cells and supporting cells. We were inspired by SYNIC algorithm, an existing method to reconstruct gene regulatory network using single-cell RNA-seq data. Based on the SYNIC algorithm, we implemented the pipeline with some differences. The two major differences of our analysis are: we integrated single-cell ataxic accessible regions in step 2, and we identified activator regulons and repressor regulons based on our TF classification results. To be more specific, in the second step, we identified putative TF binding sites from accessible regions, while the TF binding sites are within a fixed window of the TSS. And the links lacking TF binding sites got removed. Based on that, we identified regulons following by regulon activity enrichment analysis using AUC cell. We applied the 3-step gene regulatory network pipeline to hair cells and supporting cells, and generated regulon activity heat map with hierarchical clustering at single-cell resolution. Each column is a single cell, while each row is a regulon. The upper bar represents cell types and library IDs. The left bar shows the classification of the regulons. On the right, blue and yellow background indicate hair cells and supporting cell specific regulons. For instance, ATA1 and NHLH1 regulons are highly activated in hair cells, while GFI1, SOX9, and SOX10 regulons are enriched in supporting cells. To validate the gene regulatory network analysis, we ran GO-term analysis for cluster-specific regulons and compared the SOX2 regulons with SOX2 chip-seq data from an inner ear cell line. Single-cell technology reveals cellular heterogeneity underlying the developmental and spatial differences. If we concentrate on sensory hair cells only, we were wondering, can we use both datasets to reconstruct 1D spatial map along the tonotopic axis? Previous publications showed that the development of the organ of Corti precedes ingredient. For example, ATA1 starts expressing in the base of the cochlea around embryonic day 14.5, and during the next few days, it extends towards the apex. We were wondering how these gene expression gradients develop. Focusing on the hair cell cluster, we determined differentially expressed genes and differentially accessible regions by comparing cells isolated from the apex with cells isolated from the base from single-cell RNA-seq and single-cell ataxic data. We use those features to generate a 1D PCA plot, resolving individual cells' relative position along the tonotopic axis. Within the 1D spatial map, each dot represents an individual cell. The red dots come from apical compartment and the blue dots come from basal compartments. Next, we screen some candidate genes. Take PKHD1L1 as an example. Accessibility is color-coded from gray close to purple open. For better visualization, we also plotting a fitting line. Apex to base correspond to the y-axis and accessibility are represented on the x-axis. Similarly, we present the single-cell RNA-seq data with a different color code. In summary, PKHD1L1 is highly expressed and more accessible in the basal compartment compared with apical compartment. And the trend is the same for both single-cell RNA-seq and single-cell ATAC data. Our finding has been validated by RNA staining. Overall, it showed an apex to base gradient for PKHD1L1 in hair cells. In summary, we predicted the activating and repressing function of developmental TFs in cochlear hair cells and supporting cells. We inferred TF associated with downstream target genes promoting the differentiation and maturation of hair cells and supporting cells. Additionally, we resolved the original structure of the hair cells from organ of Corti along the apex to base axis and mirror the gradual changes in the epigenetic landscape during development along the tonotopic axis of hair cells. Right now, I don't have time to talk about the last session. If you're interested in it, please feel free to check our manuscript. Here, I would love to thank everyone in our lab, especially Dr. Walthouse and Dr. Liu. I'm happy to take any questions.